Now to start, Henry. Now. Just a second. Oy. Okay, so to start, Henry. Just let me turn off this. So, good afternoon, everyone. Buenas tardes. Today I have the honor to present our guest speaker, Dr. Henry McSorley from the University of Dundee in Scotland, UK. Henry did his Bachelor and Masters of Science in Immunology at the University of Glasgow, followed by a PhD in Parasite Immunology in Rick Messel's lab at the University of Edinburgh. Then he moved to Australia, where he did a research, well, he was Senior Research Officer in the Alex Lucas lab in the James Cook University and Queensland Institute of Medical Research. Then he was a postdoctoral researcher, also back in the Messel's lab in the University of Edinburgh. And he was a senior postdoc, and then he got this Chancellor's Fellowship to continue his work. And on January last year, he started his own lab, and he's currently the lecturer in immunology, as well as a joint deputy head of the cell signaling and immunology division in the University of Dundee. Henry has been granted many different awards and fellowships, including the ASMA UK Senior Postdoctoral Fellowship, the Chancellor's Fellowship, Wellcome Trust in International Strategic Support Fund, and many others. And he has currently 48 publications with more than 5,500 citations and an H index of 32. His research focuses on identifying the molecules and molecular pathways by which parasitic worms modulate the host immune system and developing this finding towards new treatments for human immune-mediated diseases. By discovering how parasites affect the host immune system, he thinks we might be able to better combat these important human and animal pathogens and learn more about the fundamental biology of the immune response. Today, Henry will talk about immunomodulatory proteins from a helminth parasite. Thank you, Henry, for being here, and we're all ears. Thanks.
Thank you very much, Paola, for that lovely introduction and for the invitation to speak. I'm really pleased to be able to speak here. So I hope you can all see my slides and they're moving. Just to confirm before I start.
Yeah, we
fine.
Thank you.
Great, thank you. It's always good to check. So I'm Henry McSorley, and I'm going to be talking about the work we do with proteins secreted by a helminth parasite, which modulate the host immune response, and how we've been using them. So just to tell you what is going to be in my presentation, I'm going to give a bit of an introduction to parasite immunomodulation in general and why we think it's an interesting area and why we think it might be useful. And then I'm going to focus on two key discoveries that we've made over the past few years. Two proteins, one called HB-ARI and the other one called HB-BARI. A lot of the work that we've done has been on using these proteins in mouse models of asthma, but then we've always tried to translate these findings from mouse models to human systems wherever possible. Although a lot of our work is in asthma and in allergic diseases, we've also tried to relate these findings back to the parasitic infections and how this could impact how we think about how parasites change the immune response in vivo. So the concept I really want to start with is that parasitic infections, parasitic worms, are really a normal part of our commensal flora. If you look at any of the oldest preserved human corpses, Egyptian mummies, corpses that have been preserved in ice for thousands of years, you'll always find parasitic worm eggs inside these bodies. And similarly, if you look in wild mammal populations, you'll find that they have very high prevalence of parasitic infection. So even if they're not currently infected, they have been exposed throughout their life. And so really our immune system has evolved in the context of this constant stimulation by parasitic worms. And in the developed world, where we have clean water and sanitation, and we've largely eradicated these types of infections, that's actually a very strange situation for the immune system to be in, as it really expects the presence of these pathogens. As a parasitologist, I'd like to make the point that not all worms are the same. And one worm which we think is particularly interesting, and really our work is based around understanding at some level, are the hookworms. So these worms here. And so hookworms live inside the small intestine, where they feed on blood by biting the walls of the gut. This is just their life cycle. Infective larvae sit on usually grass, and then will penetrate through your skin as you walk past. They'll then travel in the bloodstream and go to the lungs, where they'll be coughed up and swallowed, and they'll then mature in the gut. And in the gut, they bite the walls and drink the blood. And there they live for a very long time, laying eggs in the feces, which then completes the life cycle. And estimates vary, but hookworm species probably infect up to about 1 billion people in the world. So this is an important human pathogen. And the reason we think they're particularly interesting among parasitic worms is because there is strong evidence that they do modulate the immune response to a great degree. So if you look at most parasitic worms and lots of other types of pathogens, such as malaria, where complete immunity, sterilizing immunity, is very, very difficult to obtain, what you often get is this kind of prevalence curve, where the younger children have the highest numbers of parasites. And gradually, as over your life you obtain some degree of protective immunity, you remain infected, but at a much lower level. Well, that paradigm doesn't really exist in hookworms, where people tend to be infected over their whole lives, and there's not really any evidence of any level of protective immunity. And that implies to us that perhaps hookworm is so good at modulating the host immune response that it can completely evade these types of immune responses which are meant to kill it. So how do hookworms modulate the host immune response? Well, so this is a video of a hookworm inside a human volunteer who's been experimentally infected with hookworm. This is their small intestine, and you can see the hookworm there biting into the wall of the gut. So it's drinking the blood that way, but it's also acting like a little syringe, and it's injecting molecules into the host, and it can and we hypothesize that it's using these injections to modulate the host immune system. And so as Paula said, I trained as an immunologist initially. And so I think in terms of types of immune responses. And the type of immune response we're particularly interested in this context is a type 2 immune response, the allergic type of immune response. Type 2 immune responses are made up of various cell types and IgE and so on, which I'm not going to go into in any detail, but just to say that allergic immune responses do, of course, cause allergic diseases such as asthma. As an immunologist, that's how we tend to think about allergy in terms of these diseases that it causes. But that's, of course, not why we have allergic type 2 immune responses. We evolved them in order to combat parasitic worms, and they're particularly good at combating parasitic worms. And so if you have a mouse which lacks major elements of the type 2 immune response, then these mice are highly susceptible to parasitic infection. They really can't clear the worms. So these types of immune responses are good at killing worms, but the worms know that. The worms are very sophisticated creatures and multicellular eukaryotes. Their genomes are often about the same size as our genome. So these are complex creatures. And they've co-evolved with us for hundreds of millions of years. And as they've done so, they've developed very sophisticated ways of blocking specifically these types of immune responses. This is to their advantage because it allows them to persist within the host and not be ejected. And it also has a side effect of perhaps blocking these types of allergic pathologies. So these types of ideas are summed up in what's known as the hygiene hypothesis. This is the idea that you need certain early life infections in order to develop a healthy immune system. And in their absence, due to too much cleanliness, sanitation and so on, our immune response is set a little bit too high, and it's prone to hyperactivity and to causing these immune mediated diseases such as allergy. And so this concept really came from these large-scale epidemiological studies at first, where areas of the world where parasitic infection tends to be most common, tends to be the areas where asthma is least common. There is an inverse correlation between allergic asthma and parasitic infection. So that implied perhaps that parasites were suppressing asthma. And there's been lots of epidemiological studies in this area. And they are, of course, quite correlative, as they have to be. But this is a fairly recent study, which was a nice one, where they looked at several thousand children in Ecuador, I think, and looked at them at 3 years and looked to see whether they're infected with geohelminths, so soil-transmitted worms. So they looked at 3 years old, and then they came back when the kids were 5 years old. And stratified the children by whether they'd been infected with worms at 3 years old or not. And those children that had been infected with worms had a much lower risk of developing allergic diseases, wheeze, asthma, and also being allergic as tested by the skin prick test. So you could say that that implies that parasitic worms are suppressing the development of allergic disease. Of course, this is a correlation, not a causation. So the causation could be the reverse. It could be that those children who have a really strong type 2 immune response are really good at combating worms, so they're not infected, and they're also much more predisposed to develop things like asthma. So it's always difficult to say these studies. However, there are some interventional studies which address this. For instance, this one, where they went into an endemic population where there was lots of parasitic infections. And they treated some children with placebo control, and they dewormed other children. And then they followed them up for some years. And they found that the children who had received deworming medication developed a higher rate of allergy, as again measured by skin prick test. So that implies that, in fact, there is some causation there, and the presence of worms is required to suppress the development of allergy. In these kind of studies, they tend to take all worms as kind of one thing. So, you know, either you're worm infected or not. But of course, there's lots of different species of worms, as I've just been saying. And in this study, this is a meta-analysis where they put together all the published data and looked at different types of parasitic worm infection. And they found that when they stratified by the different species of parasites, the only one that actually gives a significantly decreased risk of developing asthma is hookworm infection. So again, just like in the infection studies where hookworm seems to be the most immunomodulatory, again, in this situation, hookworm seems to be the most interesting worm. So that's why we think hookworm is the most interesting and what we'd really like to work on. Unfortunately, there's a big problem with working on hookworms, and that is that hookworms really only infect humans. So this on the left is a phylogenetic tree of all the parasitic worms that we have good genomic data for. And in the red box there, those are the 4 hookworm species. Those species only infect humans, and also one infects dogs as well. So we want to find the closest relative to human hookworm that could be tractable for use in the lab. Unfortunately, the next closest relatives infect either pigs or horses. And again, these aren't very good lab animals. Ideally, we want something small and furry and squeaky. We want something that infects mice, basically. So in order to identify a parasitic worm which infects mice, we have to go down the arm of the evolutionary tree and up the nearest arm. And there we find 2 parasitic worms that infect mice. One's called Nipstrongylus brasiliensis, and the other one is Heligomosamoides. Nippo is an extremely useful worm. as a model of worm responses. However, it causes very acute infections that it's usually cleared within about a week, whereas Helicomosimoides forms very chronic infections in parasites. So we think this is the best model of a human hookworm. So this is what Helicomosimoides polygyrus, which is the worm we work on, what it looks like. It's a very curly worm. And it does have some notable differences to a human hookworm. It's not a blood feeder. It doesn't bite into the wall of the gut. In fact, it uses this curliness to wrap itself around the villi in the gut and hold on there. Like hookworm, it lives in the small intestine, and like hookworm, it forms chronic infections. So if you infect most mouse strains with H. polygyrus, I'll probably call it H. poly for most of the talk because it's less of a mouthful. So after you infect with H. poly, the mice usually remain patent for about 70 days or more. As a comparison, an individual hookworm in a human can last for 18 years. They're very long-lived. But again, similarly to hookworm, this is a common infection in mice, and about 80% of wild mice are constantly exposed to this worm. So just to take you through the life cycle of H. poly very briefly, and to contrast this with hookworm. So hookworm, if you remember from the beginning, it infects by burrowing through the skin, going through the lungs, and then to the gut. H. poly is again a little different in that it infects orally. The larvae are swallowed by mice. And then the infected larvae then burrow through the epithelium of the gut. So they don't then go any further. They go through the epithelium of the gut, and then they actually just sit within the gut wall in the subserosal space. So they just sit just below the epithelium, but still within the gut. And in fact, this is a picture we took very recently, which is just a dissecting microscope image of the outside of an intestine of an infected mouse at day 7, which is the time point at which these worms are in the wall of the gut. So this is from the outside of the gut. You can see the blood vessels and you can see the worms. They're very, very close to the outside of the gut, but they are still within the tissue. So they are in the subserosal space in the intestine, and there they develop. And after about 9 to 12 days, they will then emerge out. They'll burst out the epithelium, causing quite a bit of damage. And they'll go into the lumen of the gut as adults. And there they'll live for the next several weeks or months, laying eggs in the feces and living there. So as I say, it's probably the closest model we have to hookworm. There are some differences, but there's lots of similarities too. And it's been an extremely useful worm for looking at this association between parasitic infection and allergic asthma. So this is a really seminal paper in 2005, which was done in Edinburgh in Scotland, where they showed for the first time that in a mouse model, you could use a worm to protect a mouse against models of allergic asthma. So they infected mice with H. poly, and then they carried out a mouse model of asthma. And you can see one of the main readouts I'll be using throughout the talk is eosinophils in the bronchoalveolar lavage in the air spaces of the lung. And you can see you get lots in the normal asthma model, but the mice which were infected, you get much, much fewer, and the lungs also look much healthier. So this is seminal because it proved that this kind of association could be modeled in the lab, and we could look to see how this works. However, we in the McSorley lab, what we really want to do is go from these kind of phenomenological associations between parasitic infection and protection from asthma through to kind of a molecular understanding of this. So saying, you know, the reason that this worm is protecting against asthma is because it secretes this molecule, and this molecule does a specific thing. So that's where we want to get to. And the way we propose of getting there is by looking at the secretions of this parasite. So what we do is we take adult worms from the intestines of infected mice, and we can then culture them in serum-free media in the lab. For some weeks, they're fairly happy and they continue to survive, and they continue to release various factors into the supernatant, which we can then collect and concentrate. And we call these secreted factors the H. poly excretory secretory products, or HES. So these products are going to contain waste products. They're going to contain products of egg laying and so on. but there's also going to be the secreted factors, the immunomodulatory factors that we're really interested in. So it's a very crude starting point, but it's the starting point we have. When I started working in this area about 10 years ago, we wanted to see whether we could get that same association, whether we could protect a mouse against a model of asthma using just the secretions, not the whole infection. So here we're using a slightly different model of asthma, which is dependent on Alternaria allergen, a fungal allergen, delivered into the lungs of mice, and you can then recall a response a couple of weeks later. And we administered HES just once at day 0. And similarly to the infection data, you can see that you get good eosinophilia in the positive control mice, and that is very significantly suppressed when those mice had received HES. And again, the lungs look much healthier. So you can see here, there's lots of inflammation in the positive control, and the mice which received HES, that is a much healthier looking lung. And to cut a very long story short, we then looked at really the very earliest events in this model. So within an hour of administration of Alternaria to a naive mouse, you get release of this particular cytokine, IL-33. And it had already been known from the literature that IL-33 was really critical for this model of asthma. And we found that the mice which had received HES released less IL-33, and that seemed to be responsible for all of the suppression downstream. So something in HES is suppressing the release of IL-33. And so this work really started about 2010, and at the same time, there was the first genome-wide association studies of the heritability of asthma. And these studies showed that asthma did have a genetic basis at some levels, and that there was a kind of handful of genes only, and not that many genes which were associated with asthma. But 2 of those genes were IL-33 and the IL-33 receptor. So this is really exciting because it implied that something in the worms was modulating this pathway, which had just been discovered to be really important in human disease. And so lots of people were then looking at how we could block the IL-33 pathway to treat asthma. And as I'm saying, we had something from the parasite secretions that seemed to be doing that. So just a little bit about IL-33 as a cytokine, because it's quite an unusual cytokine. IL-33 is not conventionally secreted. It's known as an alarmin cytokine. It's constitutively expressed in the epithelium of the lung and other sites in the body. And there it's stored within the nucleus, bound to chromatin. So it just sits there in the nucleus, it's kind of stored there. And it can't be really released by any conventional means. The only way that it can get out is if that epithelial cell undergoes necrosis. So if something really nasty is introduced into the lung, such as Alternaria allergen, which is quite toxic. So you get necrosis of the epithelium. That leads to release of the nuclear contents, release of IL-33, and IL-33 can then bind to its receptor, which is called ST2, and a whole range of different immune cells, which are very pro-allergic immune cells. And they'll go on to induce the kind of pathology associated with asthma. And what we were finding was that secretions of our parasite were able to block that early IL-33, and that was blocking everything downstream, and the mice weren't getting the pathology associated with asthma. So of course, the next question was, well, what is it in that mixture of secretions which is blocking the IL-33? And so in this talk, in the interest of time, I'm not going to go into how we identified this, but basically we whittled it down to a smaller and smaller number of molecules until we identified a single molecule, which we called the alarmin release inhibitor, or ARI for short. And ARI we can now produce as a recombinant protein and show that it can act against the IL-33 pathway. So this is data I just showed a second ago, showing that HES can suppress the release of IL-33 induced by Alternaria. And what we can now do is, again, at these very early time points, we're now looking 15 minutes after Alternaria administration, we can again show lots of IL-33 released, and ARI is able to block that extremely effectively, better even than HES. We can also go into the kind of full model of asthma, the 17-day long model of asthma, administering ARI just once at day 0. And again, we can see that these lungs look much, much healthier. There's hardly any eosinophils there, and the whole immune response has been suppressed in this model. Importantly, for kind of clinical relevance, we wanted to look at lung function, which is obviously important for clinical asthma in people. And so in these mice that had received ARI, they had reduced resistance in their lungs and increased compliance. And these are good. That means that the lungs are working better. ARI is very able to suppress in this Alternaria model of asthma. However, I mean, one criticism we occasionally get is that the Alternaria model of asthma is highly IL-33 dependent, and it's quite an unusual model. It's maybe not the most clinically relevant. So what we've been doing recently is using another model, the house dust mite model, which the house dust mite allergen is something that a lot of human asthmatics are allergic to. And so it's probably the most clinically relevant model. And so what we've been doing is using ARI in this model too. And again, we can see good suppression of eosinophils. We seem to be getting some suppression of the kind of type 2 immune response in general, but we've clearly got more work to do here, but that's what we're working on at the moment. So ARI is able to block responses in this mouse model, in these mouse models of asthma. But of course, we next wanted to know how it was doing that. How was it acting against the IL-33 pathway? How is it blocking its release? So we first thought, well, maybe ARI is itself binding to IL-33. So we used an immunoprecipitation approach to see whether we could see the interaction between ARI and IL-33. We used MICTAG ARI, which we could then pull down using an anti-MIC antibody on magnetic beads. And we mix this with mouse lung homogenate. So as I say, IL-33 is constitutively expressed. So if you homogenize the lung, there's lots of IL-33 in there. And then we could then pull down ARI, and with it, we pull down IL-33, as you can see here. So indeed, ARI does bind to IL-33. We were surprised to find it also has a second function, which is it also binds to DNA in a kind of non-sequence specific manner. So here is a very similar experiment using immunoprecipitation, where we're just pulling down plasmid DNA, so just DNA from bacteria. And you can see, again, ARI is able to pull this down very effectively. So it binds both IL-33 and DNA. Why would it want to do that? Well, here's our model of what is happening. So you've got the IL-33 within the epithelial cells of the lung. And when they necrose, they release their stores of IL-33, and that acts on its receptor. ARI is able to bind directly to the IL-33, and we know from binding studies that binding to IL-33 prevents IL-33 from binding to its receptor. So in that sense, ARI is acting a bit like a blocking monoclonal antibody. However, the second thing that ARI does is a bit cleverer. So what ARI can do is it can also bind using its first domain to genomic DNA. And so it uses the genomic DNA as kind of like a substrate. It grabs onto it, and it grabs onto IL-33 with its second domain, and it tethers the IL-33 within the dead cell, preventing the IL-33 from ever being released. And that makes it an extremely effective blocker of IL-33 responses. And just at this point, I'd just like to highlight that ARI is a 28 kilodalton protein. It can't cross cell membrane, so it can't get into a live cell. The only time it can get in and bind to that DNA is when the cell is already lysed and the membrane is ruptured. So that was, an interesting story. But what we really want to do is see whether we can use this knowledge to develop new therapeutics for human disease. Can we come up with a new drug for asthma from this? And so what we decided to do is take an approach where it would be nice to have the minimal size of molecule possible, because that might make it a better drug, easier to produce, and so on. So we have this molecule which has 3 domains and which binds DNA and IL-33. And we started doing some truncation studies to look at what the different domains did. And we made this truncation, which we call CCP12, which is just the first 2 domains of ARI. And we found that CCP12 is still able to bind both DNA and IL-33. So this is a gel shift assay for looking at DNA binding. You see ARI binding to DNA and inducing a shift up the gel of the DNA, and CCP12 doing the same thing. When we make a different truncation of CCP23, we find that it can't bind at all. So the binding is through the first domain. Using Biocore surface plasmon resonance, we can show ARI binding to IL-33 with about 1 nanomolar KD, so very strong binding. And our truncation mutant still binds to IL-33 very strongly, about 10-fold weaker than the original molecule, but 10 nanomolar KD is still pretty high. That's pretty good binding. So we thought it would be worthwhile investigating this molecule further and seeing whether it would be a decent therapeutic. And so we looked at our very early release of IL-33 after Alternaria. Again, this is similar to previous data. You can see that ARI was blocking that IL-33 release at these very early time points. And when we use CCP12, it was really almost as good as the original molecule. So again, we thought, okay, well, let's see if it can suppress in the whole mouse model of asthma, 17-day long model. So of course, because nothing is ever easy, it doesn't work at all. In fact, it does the opposite. So CCP12, rather than suppressing these responses, as you can see with ARI, actually amplifies IL-33 responses. So you get increased eosinophilia in the lung and increased responses of IL-33 responsive cells, ILC2s, as shown here. And so this was really an enormous response. This is the largest ILC2 response we've ever seen in a mouse. So CCP12 was certainly very potent, but it was doing actually the opposite of what we expected it to. So we wanted to look at, this wasn't what we wanted, but we still thought it was interesting enough to investigate further. So we can look at an earlier time point. So this is just looking at 1 day after Alternaria administration, where you get a purely innate response to the allergen. And what is nice at this time point is you can get an eosinophilic response to Alternaria, as shown here, and you can block this with ARI. And this is a high dose of Alternaria, which is what we use normally. But what we could also do is use a low dose, where you get an almost negligible response to the Alternaria allergen. However, when we co-administered CCP12, suddenly you get a really decent allergic type of response. And again, these ILC2s, these IL-33 responsive cells, were responding very strongly in the presence of CCP12. And it's not just in these Alternaria-dependent models. We've also used this other parasitic worm, Nipstrongylus brasiliensis, and shown that while ARI can block the responses to this worm, CCP12 amplifies them. And you get more type 2 associated cytokines, IL-5 and IL-13, in the presence of CCP12, and more eosinophils as well. And then this is also a kind of a type 2 immune response, but we wanted to look in a more inflammatory situation. And so what we can do is inject IL-33 into the peritoneal cavity. And if you do that, you actually get a neutrophilic response, a much more inflammatory response, not so pro-allergic, which is, again, obviously IL-33 dependent. And we get the same results again. So you give IL-33, you get lots of neutrophils, you give ARI and you don't. But if you give CCP12, you get tons of neutrophils, sorry. So whatever the situation, CCP12 seems to be able to amplify the response to IL-33. So we wanted to see if we could find a mechanism by which this molecule, this new molecule, was working. So we tried to create an in vitro assay to kind of model this. So this is quite a complicated assay for us anyway. So I'll just take you through it. So we had an epithelial cell line which expresses IL-33. And what we do is we'd freeze thaw it to induce necrosis of all the cells. Once those cells are thawed, you get loads of IL-33 released into the supernatant, all those preformed stores of IL-33. And the thing about IL-33 is it's a very labile cytokine. And so it's very rapidly inactivated once it's released. So we then kind of incubated these supernatants for a time course up to 24 hours after we'd thawed them. And then what we do is we take these IL-33 containing supernatants and add them to bone marrow ILC2s, again, this immune cell subset, which is highly responsive to IL-33. And we measure the response, which is IL-5 and IL-13. And what we found was that in the media control wells after the thaw, if you added those supernatants after an hour or two, you got a decent response in the ILC2s. So that IL-33 was able to stimulate the ILC2s, and you got IL-5 and IL-13. But very rapidly, if you continued incubating those supernatants before adding them to the ILC2s, it lost its effect. The IL-33 became denatured. And we know that this response is IL-33 mediated, because if we add ARI when we thaw the cells, then you don't get any response at all. So again, so this is an IL-33 dependent response, but it's very transient because the cytokine is so labile. When we added CCP12 to the well at thaw, what we found was we got a 10-fold increase in the response of the ILC2, so much, much more activated. And this response never went away. So the IL-33 seemed to retain its activity even after you'd kept it at 37 degrees for 24 hours here. We've actually rerun this experiment out to about a week, and the IL-33 stays fully active. So CCP12 seems to stabilize IL-33, and it seems to be doing that in vivo. So it binds to IL-33, but it doesn't block its activity. And it just keeps the IL-33 in its active form, and that can really amplify the effects of IL-33 in a number of different situations. So just to highlight again, the CCP12 truncation mutant that we made is not a natural molecule. So this is a purely synthetic molecule which we made in the lab. It's not produced by the parasite, but it's been a really useful research tool. So we can use it to amplify IL-33 responses, and we can use the original ARI molecule to suppress IL-33 responses. So if anyone's interested in working on IL-33, we're more than happy to send this molecule to you. So that was a couple of stories all about the ARI protein, which we identified from the worm. For the rest of the talk, I'm going to talk about a second molecule, which we kind of discovered off the back of this work. So this really stemmed from this observation here. So what we're now looking at is SG2, which is the IL-33 receptor on the surface of these ILC2s, which I keep on mentioning. And if you give Alternaria, you get an IL-33 release, and that IL-33 release upregulates its own receptor. It binds to SG2 and then it upregulates SG2. And so you can see in Alternaria, you get increased SG2 expression by mean fluorescence intensity. If you give ARI, you block the IL-33 response, and it just goes back down to baseline, as if the mice hadn't received Alternaria. So that all makes sense. But what we were really surprised to find was that when we gave HES, the entire secretions of the parasite, which we started with, it seemed to completely ablate the signal that we get from SG2. So it was like SG2 had disappeared off these cells. So that implied to us that there was something else in HES, something that wasn't ARI, that was somehow blocking the IL-33 receptor. So just back to the schematic, we've got ARI, which is blocking IL-33 in this model. And we've got something else from these secretions, which is blocking SG2. And again, I'm not going to go into how we identified this. This is all published if you're interested. But we identified another molecule, which we called binds alarmin receptor and inhibits, or BARI. So BARI acts on ST2. And BARI is also an extremely effective molecule at blocking these types of responses. So you could administer BARI to a mouse, and you can block the eosinophilia to our Alternaria allergen again. And what's more, you can also get the same effect, which I just showed with HES, this disappearance of the IL-33 receptor, ST2. So if you administer BARI alone or with Alternaria, then the signal for the ST2 really disappears. And these are some flow cytometry plots, which is actually the raw data from which we got this. And so our IL-33 responsive cells lead lineage negative ST2 positive cells. So here you can see that signal. You can see in the presence of BARI, that signal really profoundly disappears. So we hypothesized that BARI was doing something directly to the ST2 protein, a bit like ARI was doing to IL-33. Indeed, that does seem to be the case. So we can carry out an ELISA where we coat a plate with either BARI or ARI as a control, and then we can use a soluble form of ST2, and we can detect that, and we can see that interaction very strongly with BARI, but not with ARI. So BARI does bind to ST2, and again, we can use surface plasmon resonance to measure the affinity of that interaction, and it's again very high affinity interaction. So this is a sub-nanomolar KD, which is very strong binding. So BARI acts by binding to ST2 and blocking it and preventing IL-33 from interacting with its receptor. And in a completely kind of separate way, it blocks the IL-33 pathway from ARI. So again, this is really interesting. It's nice to see that we found these parasite proteins and so on. But the whole reason we kind of started this research was to model human hookworm. And the whole way that I'd always envisaged this kind of work was that we were going to identify the immunomodulatory proteins from H. polygyrus, our model parasite, and then we're going to find the homologous proteins from the human hookworms, and those were going to be the more interesting molecules to work on because those are adapted to the human rather than to the mouse. Unfortunately, when we actually take the sequences of Ari and Barry and compare them to the hookworm genomes, and in fact any of these parasite genomes here, we can't find any significant homologs. There's nothing really there with any significant level of homology. So this seems to be, strangely, something which is unique to H. polygyrus. We don't think it's unique in the sense that I'm sure that other parasites are blocking the IL-33 pathway. It's clearly important in anti-worm responses, but they seem to have a different solution to that question. So Ari and Barry seem to only be in H. polygyrus. But what we did find when we were comparing these sequences were there was a number of Ari and Barry homologs present in the H. polygyrus genome. So we thought it might be worth looking at as well. So we're wanting to translate these results to humans. So again, this is just some binding data, looking at the affinity of binding of Ari to IL-33 and Barry to ST2. And like I say, both of these are very high affinity interactions. But what happens when we swap out the mouse version of these proteins for the human version? So if we look at Ari versus human IL-33 and Barry versus human ST2, we get a much, much decreased affinity for the human versions. And that makes sense. It's disappointing that it makes sense. Ari and Barry were evolved to combat the mouse immune system, not the human one. So it's not that surprising that they bind less well. However, we've got these homologs of Ari and Barry in the H. polygyrus genome, so we thought it might be worth testing them as well. So there's several homologs of Ari and several homologs of Barry. And so we're testing here Ari3 versus original Ari and Barry Hom2 versus original Barry. And what we found first off is that Ari3 and Barry Hom2 are perfectly able to act against the mouse version of IL-33 or ST2. They bind with high affinity still. But very excitingly for us, they bind just as well to the human version as they do to the mouse version. So I have no explanation for this whatsoever. This is a mouse parasite, and yet it's producing a homologous molecule to Ari and to Barry, which has really strong activity against the human version of IL-33 and ST2. And again, these parasites never infect humans. So I don't know why they're there, but it's great that they are there. And what we can do is we can take these homologous proteins, Ari3 and Barry Hom2, and use them in human assays. We've recently used Ari3 in a human IL-33 assay. I'm afraid I can't really show you that today, but what I'm showing you here is Barry Hom2 in a human assay. So this is a human assay where you take human PBMCs, you stimulate them with IL-12, and that upregulates the IL-33 receptor. You then stimulate them with IL-33, and you get interferon gamma as a readout. That is shown here. And what we could do in this assay is we could titrate in either original Barry or Barry Hom2, and we can see in blue that Barry Hom2 is really very good at blocking human responses to IL-33. So it does seem to be a functionally active molecule. And we're looking into this further, obviously. So there is potential for these molecules being translated towards human treatments, towards treating human allergic disease. Another kind of thing you have to think about in developing these kind of drugs, I guess, is how do you deliver these molecules? So all of the data I've shown so far, we've been delivering Ari or Barry directly into the lungs of mice by an intranasal route. You can do that in humans. You can deliver proteins into the lungs of people, but it's not easy, it's not cheap, it's not really the way you want to go. So we wanted to see whether you could deliver these proteins systemically, so either intraperitoneally or intravenously, and could you still get a suppression of responses? Frankly, I didn't expect this to work because I thought that these proteins would have to be at the site where the IL-33 was released. However, to, again, my surprise, both Ari and Barry work any one of these administration routes, so intranasally, intravenously, or intraperitoneally. Intraperitoneally, incidentally, it seems to work the least well, but it still works pretty well. So this is eosinophil responses to Alternaria, and then we've got activation of our ILC2s, or IL-33 responsive cells. And I just find this quite a nice bit of data. This is ST2 expression on our ILC2s. This is the IL-33 receptor. And here you can see the really stark difference between Barry and Ari. So Barry completely ablates the signal for ST2, whereas Ari really does nothing. So we can give these molecules by a number of routes, and they should be able to systemically block IL-33 responses. The next thing that we wanted to do with these was see what they were doing actually in the infection with the parasite. So we know that they can act at different sites. So does that also hold true for the parasite? Is the parasite releasing these molecules and they're acting systemically? This is just a diagram of a mouse, and the parasites are here in the proximal duodenum in the small intestine. And you're going to be looking here at day 7. So this is this time point when the parasites are buried in the walls of the gut. And the first thing we did, this is work done by Josh in the lab. He looked at the peritoneal cavity, which is obviously just next to the intestine, and looked for ST2. And so he's looking for ST2 suppression as a kind of proxy of the effects of Barry. And you can see that ST2 here in peritoneal immune cells, ST2 is very potently suppressed. So it does seem to be able to act outside the intestine, at least in the peritoneal cavity. He then looked further afield. He looked in the lung, because we've obviously got an interest in the lung, and again saw potent suppression of ST2. And most recently, what he's been doing is looking in the fat. And again, IL-33 is a very important cytokine in the fat. It controls metabolism. And he looked for the expression of ST2 in fat immune cells, and again, really, really potent, expressed, virtually absent. So despite this worm being kept within the intestine, its effects on ST2 are systemic and a lot wider ranging than I initially expected. We've got another PhD student, Nicole, and she's been looking over a kind of time course to see how long these effects are maintained. So just a very quick reminder of the life cycle of this worm. You get infective L3s coming in. They sit in the wall of the gut for about 12 days, and then they emerge into the lumen of the gut as adults from 12 days onwards. So she looked at the suppression of ST2, again, as a kind of proxy of Barry's effects. And this is just looking at the peritoneal cavity here. And she found that really that only happens at day 7. So only when the parasite is within the wall of the gut, so you get this really profound suppression of ST2. And as soon as she goes to day 14, when the parasite is in the lumen of the gut, that suppression disappears entirely. So there seems to be something really important about this tissue-dwelling phase of the parasite that kind of modulates the immune response, and we hypothesize changes the immune response downstream in a lasting way. So that's me. Just to summarize, we found several immunomodulatory proteins from this parasite. We think there's much, much more to be found, and we are now finding other immunomodulatory functions of the secretions, and we're finding new hits, which hopefully we'll be talking about in future talks. And we think that these immunomodulatory factors secreted by the parasite could have potential as new treatments for allergic disease. Obviously, I'd like to thank everyone who actually did all this work, everyone in the lab, as you can see here, the University of Dundee and our funders from Longfons, Netherlands, the Wellcome Trust and the MRC. And just as I finish, I'd like to say we've got a postdoc available in the lab to work on the asthma and helminth product work. So if you're interested in that, please do contact me. And I will finish up there. Very happy to take any questions you have at all.
Joe, Paola? All right. I'd like to know whether you have tested other self-allergy reactions like arthritis, lupus.
Yeah. Yes, so I haven't personally. My first postdoc was on inflammatory bowel disease, on colitis, where parasitic secretions do show some effects. We've stayed largely within the type 2 immunity, so asthma, allergy, atopic dermatitis and stuff like that. But there's lots of people working on how worms suppress multiple sclerosis, arthritis, as you say. And it seems to be through different pathways. So part of the thing that worms can do is induce type 2 immunity, and type 2 immunity will then suppress type 1 immunity, and type 1 immunity is largely what causes arthritis and things like that. So there's some things that worms do that are as a consequence of the immune response against them, and some things which are due to immunomodulation, and I think both are happening. So there's a lot more complexity there. But yeah, so not something we've done personally though, so I can't answer that very
right. And then, if you inhibit this type 2 response, aren't other infections promoted within the animal, like other worms or helminths?
Yes, absolutely. Yeah. So there's a lot of work ongoing on kind of co-infections between other worms, and how immunomodulation by one worm can affect the response to the other worm. Quite often it seems that the immune response is primed by one worm to eject the second worm, and perhaps that would be in the first worm's interest to do that, because it doesn't want someone else coming in and stealing resources. But yeah, there's lots of evidence that parasitic infections can then decrease the efficacy of vaccines, things like BCG, and also can suppress the response to bacterial and viral infections. So yeah, so that co-infection field, again, very complex and very interesting.
Thank you.
Thank you.
David.
Sí, gracias, Paula. So thank you very much for your work, Henry. I have a couple of questions. Do you know if these domains of this ARI protein you have could bind some other targets?
Yeah, I mean, I think that's a really interesting question, and I really want to find out the answer. So we're trying to do some more kind of unbiased approaches at the moment, seeing if we can use things like that immunoprecipitation technique and other kind of binding studies to see whether we can, whether ARI is binding to anything else and pulling down anything else. And the same thing with Barry and other parasite molecules. And again, kind of preliminary data indicates that yes, I suppose, unsurprisingly, when you think about things like viral immune evasion proteins, which often have multiple effects within a single protein, yeah, these types of proteins can bind to other stuff as well. With ARI, I don't think we found anything convincing yet, but we're certainly trying hard with that.
Yeah, well, that is, I mean, a possibility that stems because when a cell just breaks, there are so many things that can elicit an immune response that it is surprising that just the binding to IL-33 will block it. So, and indeed, it's very interesting that this mechanism seems to be redundant, because there is also this body thing that in both, yeah. It's very, very nice work. And the other thing I was willing to mention that perhaps may help you to think about it, it's with a three-point bridge, you can do a network. With a two-point bridge, you do but chains. So I don't know if this 3D organization between ARI with the two domains or the three domains just change it, because how it is being oligomerized IL-3 or whatever it binds, right?
Yeah. So we know that well, at present our best data says that ARI exists as a monomer and acts as a monomer, whereas Barry actually exists as a tetramer, and so there could well be grounds for thinking that. Indeed, just I'd like to answer the other part of your first question as well, if I may. So you were saying lots of stuff were released from necrotic cells, and absolutely. So some important things are DNA and ATP are both extremely inflammatory, released from necrotic cells, and they can really help to set off the inflammatory immune response. Our parasite secretions also, we know, contain DNases and apyrases, and DNases obviously degrade DNA, and apyrases degrade ATP and render it non-inflammatory. So again, I think it's probably, I've presented this story in IL-33, and I think there's probably a lot to be said for you find what you're looking for. And we're using a model of asthma, which is highly IL-33 dependent. So we found a lot of stuff to do with IL-33, but there's certainly lots more stuff out there. which we just haven't found because we weren't looking for, I think.
Yeah, it's also very interesting because IL-33 has been found in the brain to regulate through the activation of microglia, the neuronal circuits. It's quite interesting. Thank you very much, Henry.
Thank you.
Una pregunta. Thank you so much for your talk. Can you tell us a little bit more about the protein itself, about its structure? I mean, do you find zinc fingers or?
Yeah. So funnily, strangely, both Ari and Barry are structurally related, even though they have separate targets. So they both consist purely of CCP domains with nothing else. So these are complement control protein domains, which are found in the complement system, but also they're involved in loads of other things, neurotransmitters, all sorts. These are generally about 70 amino acid domains, and they have 2 disulfide bonds within them, which makes them very stable. These proteins are all, you can leave them at 37 degrees for weeks on end and they're completely fine. But they're also kind of modular, so you can join together lots of CCP domains. And so, for instance, factor H, which is part of the complement system, consists of about 20 CCP domains. And you can say these 3 do one thing and these 2 do another thing. And so I think there's quite a lot of scope there for the parasite to be able to kind of use them like Lego and build what they want. And we also know that H. polygyrus in its genome, it's had this massive expansion of the CCP domain family. And it again seems quite unique in that. So we think there's that's kind of the answer that is chosen, this kind of quite stable protein domain family. But yeah, again, very interesting question and one we want to look at further. Thank you.
Thank you.
Feliz, ¿tenías una pregunta? Feliz.
No, yo no tengo pregunta.
I have a question, Henry. So you said you didn't found any like similar molecules derived from the human parasites, right?
Yeah.
But I was thinking if you were maybe considering using a different approach, just to kind of like make these extracts from the worms, from human worms, and then catching those, like use them as a how do you say like a trap to interact with IL-33 or any other like alarmine or molecule relevant for type 2 immune responses. If you're planning on doing something like that, but because I mean, it's very amazing. I love how you found these molecules and how the parasites are able to modulate and control the immune response. That's amazing. And I found hard to believe that this same thing cannot happen in human parasites, which we also have like years and years of evolution together, and we must have some ways to have the same type of these worms must have similar strategies to avoid the immune system. So probably we were planning on using different approach. And my other question is, how does ARI binds to DNA?
Okay, so first question, which I've actually got an answer for. I'm not sure I have an answer to the second one. Yes, so the problem, we'd love to do that. We'd love to take the secretions of the human hookworms and look for those IL-33 binders. And I really like that, your idea of using them as a kind of trap and fishing them out. The problem is that it's almost impossible to get the adult worm secretions from the human hookworms because we have to kill the mice and pull the worms out of the small intestine to get the adults, and so there's not really any way to access the live adults from the human hookworm infection that we can think of, and we need them live.
With egg extracts?
Well, I mean, the thing about the egg, we've done some work with the egg extracts with H. poly, for instance. The eggs don't really have much of a reason to want to modulate the immune response. They actually want to be ejected as soon as possible. So we don't think the egg extracts in these cases are very immunomodulatory. In things like schistosomes, they are, because the eggs are actually buried within the tissue. But in these ones, they just need to make their way out in the feces, basically. But yes, no, it would be great if we could grow a bunch of human hookworms and do that. There are some people who have managed to make some human hookworm ES, secreted products. But it's gold dust, basically. But yeah, we'd love to try that, and I think that's a really nice idea. And then your other question was how ARI binds to DNA. Yeah, we really don't know. We've done some molecular modeling of the protein, and specifically of that first domain. The CCP1 domain is the one that binds to DNA, and it seems to have exposed basic patches on its surface, which in other CCP domains, it's been shown that they can electrostatically basically interact with acidic DNA, and so it's certainly not a sequence-specific interaction. It's just any DNA. So we think it's probably just an electrostatic interaction, but yet we don't know in any detail.
And also, just one more thing, coming back to this molecule you found that binds human ST2 or IL-33, and you said you were kind of shocked because they were crossing between species and stuff, but maybe once you map the binding site of these molecules, then probably they will be hitting on sequences that are conserved between human and mouse. Have you looked at this? Like, what's the homology between IL-33 in human and mouse?
Yeah, so IL-33 between human and mouse is surprisingly low homology, actually. I mean, it's still homologous, but both proteins have got kind of, I think either 4 or 6 cysteines, but they're in entirely different parts of the molecule. So it's quite low homology. But yes, I mean, I what we're trying to do at the moment is we're trying to crystallize the IL-33 ARI complex and then get a crystal structure of it and then find out where those binding sites are, because we don't know at all where ARI and IL-33 are interacting well on the proteins. And knowing that kind of information, exactly as you say, that would make us be able to understand that interaction much better, but we don't know yet.
Okay, I have one question from the chat. Have you ever checked the effect of HPRI over IL-9 expression? Do this cytokine precedes and its upstream of expression of 5 and 13 in the model of Mypostrangulus?
So we so it doesn't seem to bind to IL-9 because we have actually tested that. However, we just published recently with Minka Brola in Hamburg, and she is working on strongyloides where the ejection is extremely IL-9 mast cell dependent. And what she found was that that IL-9 was induced by IL-33. So there's an IL-33 IL-9 mast cell axis. And she, in fact, in that publication, she used ARI and could show that she could block the ejection of strongyloides. So not exactly the experiment you're saying, but yeah, on a similar theme, I would say.
Okay. Well, I don't know if there's any other questions. Eugenia.
Yes, thank you very much, Henry, for your talk. So I was wondering, in the long-term use of HPR proteins for asthma treatment, as this is a foreign protein, at the end, the host will elicit an immune response, the production of antibodies. So do you expect that we could use this protein in a long-term use?
Yeah, I'm afraid you've put your finger on a really sore point. It's a real problem for developing these proteins as therapeutics, is that they're entirely foreign, and so they could be immunogenic. It's not a terribly satisfying answer, but we have shown that for instance, in actual parasite infection, you get an antibody response which binds to ARI and BARI, but it's completely unable to block their effects. So even if they're immunogenic, that might not stop them from working. But it's been enough so far to discourage industrial investment in developing these as therapeutic molecules, which is disappointing, but I can understand why. Another kind of approach which we're hoping might still be useful in that kind of therapeutic realm is that these parasites do seem to have fairly unique ways of blocking the immune response. And if we can work out those molecular pathways, for instance, that dual IL-33 DNA binding of ARI, if we can, mimic that using non-immunogenic proteins, or come up with a strategy which kind of replicates what the parasite is doing rather than using the parasite molecules, then maybe this could still be used therapeutically. But you're absolutely right, it's a big problem.
Thank you.
Thank you.
Okay. So I don't think we have any more questions. Thank you very much, Henry. It was nice to have you. And I don't know, thank you everyone for being here.
Thank you very much for the invitation. It was great to speak. Thank you.
Thank you.
Henry, thank you very much in the name of the Institute, and thank you for being here with us.
Thank you very much for the invitation.
Have a nice weekend.
Yeah, you too. Thank you. Bye-bye.
I'll email you, Henry.
Gracias, Paula. Buenas. Y saludos a todas y todos. Buen fin de semana. Gracias.
Gracias, igualmente.
Gracias, igualmente.
Gracias. Hasta luego.
Hasta luego.